We don't have many participants, but let's begin. Are there any questions? So today we're going to cover Lab 6. Please read the unknown projects file on Canvas. I'm going to talk about the unknown project next week. That means I got to send out the unknown project. Okay. Oops, what's going on here? Oh, there it is, all right. Can somebody confirm that I have the, oh, there we go. Yeah, I am recording, all right. So, Lab 6, Dist Diffusion, evaluation of disinfectants and antiseptics, and antibiotic effectiveness. Please do the reading, which is sort of scattered throughout the textbook, but do redo the reading. Watch the video clips and do the virtual lab. Learn your learning objectives. Identify the chemical agent and active ingredient used in various disinfectants and or antiseptics. Evaluate the effectiveness of disinfectants or antiseptics using the Dist diffusion method. Determine the mode of action of various antibiotics. Evaluate the effectiveness of various antibiotics. List the variables that influence antibiotic Dist diffusion susceptibility testing. Particularly pay attention to that one. And then know the terms: sterilization, disinfection, disinfectant, antiseptic, bactericidal, bacteriostatic, antibiotic, a spread plate, and zone of inhibition. We will use the term antibiotic as it is commonly used. Textbooks tend to use the term antibiotic to mean a antibacterial drug that is made by another living organism, and we will use the common term for an antibiotic, and that is an antibacterial drug that is either made by man or by a living organism. For example, in the textbook meaning of the term antibiotic, penicillin G is an antibiotic because it's made by the penicillium mold, but carboxycillin is a man-made penicillin, and so by that term it is not an antibiotic, and by common term it would be an antibiotic, and so we'll use the common term. Any questions about any of that? So in the lab, you will be evaluating disinfectants and antiseptics. Learn what sterilization is, disinfection, what disinfectants are, and how they differ from antiseptics. A disinfectant is a chemical agent applied to an object. An antiseptic is a chemical agent applied to living tissue. So if you use bleach to clean your toilet, it's a disinfectant. But if your cat comes home scratched up and you put bleach on the cat, which most people wouldn't do, but if you do, you'd be using bleach as an antiseptic. Is that clear? Learn how to evaluate the chemical agents. We apply them to the surface of a nutrient agar plate that's inoculated with a bacterium. And usually you have the bacterium on the plate as a lawn of bacteria, meaning it'll grow up everywhere. And then you put the disinfectant or the antibiotics on the plate. After you put your bacteria everywhere, you put the antibiotics or disinfectants in a paper disc, and then you put that paper disc on the plate. The chemical agent then diffuses into the agar medium and establishes a concentration gradient around the disc. So it's highest near the disc, and as you go out further, it gets lower. And you'll see around the disc, if the bacteria is sensitive to that agent, you'll see a zone of inhibition where there is no bacteria growing around the disc. Now, when you look at that, the bacteria may be inhibited or they may be killed. And when you look at it, there's no way to tell the difference. Any questions about that? Does anyone have a guess how you could tell a difference? That's actually one of your questions in the lab. Well, think about it.
I hope it's not too windy, but I was just thinking if it was an inhibitor that you would still have some that are working, where the ones that is more clear.
So you're saying that the one that kills the cells will be a total clear zone of inhibition, and one that inhibits will not be totally clear. You'll see some haze in it. That's a good guess, but no, that's not the case. Because the bacteria are not visible to our eyes if they're not growing. Anyways, think about it, and it'll be one of the questions. I'll get back to you on that when we're answering the questions. If there is no zone of inhibition around the disc, that means that bacteria is resistant to that agent in the disc. Okay? Any questions about that? Now, whether something is sensitive or resistant is not dependent on whether there's a zone of inhibition or not. And that's a little bit more complex, and I will eventually talk about that. So hold off on that discussion. I know I just confused you, but I'll briefly state it and we'll go over it again. And that is, if you have a zone of inhibition, it doesn't mean the bacteria are, quote, sensitive to the agent in that disc. You have to measure the zone of inhibition and then look it up on a chart. And if the zone of inhibition is greater than it says on the chart, then you say that that bacteria is sensitive to that agent in the disc. Okay? And if it's less than a size in the chart, then you say it is resistant to that agent. And so you can have a zone of inhibition, let's call it small, and the agent is resistant to the agent on the disc. Did that make sense? So you have to look it up in a chart, and you have to measure the zone of inhibition before you say whether it's sensitive or resistant. And we'll talk about that more later. So you should refer to this table. I'm not going to ask you to memorize it, but there are different agents, the chemical agents, alcohol like ethanol and isopropanol. They denature proteins and dissolve lipids, and we call them an antiseptic or a degerming agent because they're not real good at killing, but they're okay, and we use them on the skin to like degerm a region where you're going to stab a needle into a patient, but it's not really a strong killer, isopropanol and ethanol. So you should refer to this table. I'm not going to ask you to memorize it, but you should look at the table and know how to use it. I don't think I'll even ask you questions on a quiz about it. If I were to ask you a question about it, I will supply this table. So you need to know how to use this table. Any questions about that? Later on in the term, we'll talk about disinfectants and antiseptics. It's chapter 7, in which case I might be a little more specific. But for now, just know how to use this table. I think I already talked about that. Oh yeah, the zone of inhibition size depends only partially on the sensitivity of the organism to the disinfectant or antiseptic or antibiotic. There are other factors involved determining the size of the zone of inhibition. That's why you can't simply just look at it. And that is, the agent has to diffuse through the agar. And the smaller that agent is, meaning a smaller agent, will diffuse at a greater rate through the agar than a larger agent. And that's why you just can't look at the size of the zone of inhibition and determine whether it's sensitive to the agent, the bacteria, or whether it's resistant. You have to measure this zone of inhibition and look it up in a chart. Because of the fact that the agent is diffusing through the agar, when we do a test like that, you have to standardize a lot of the variables so that you will get similar results each time you do the test. Any question about that? All right, evaluation of antibiotic effectiveness. You can take, let's call it not chemotherapeutic agents, but antibacterial agents, and use them to treat against bacterial disease. And so when a patient has a bacterial disease, and regardless of whether you know which disease it is, you might want to ask what agent will work good on treating that bacterial infection. And what you can do is take that infection and grow it up on a petri dish, and then apply different agents on a disc and see how well they respond to the bacterial agent, meaning do they have a big zone of inhibition, a small zone of inhibition, or no zone of inhibition. And you can do that to test for the effectiveness of antibiotics. It is true that the antibiotics have to diffuse through the agar, but that's very similar to the way that when you give a patient antibiotics, how the antibiotic has to diffuse from the blood and into the tissue. And so this evaluation method is called the Kerby-Bauer test, where you put antibiotics on a paper disc and then allow the antibiotic to diffuse through the agar and then measure the zone of inhibition, is actually a very good test for determining the effectiveness of an antibiotic in a patient, because the antibiotic is going to be diffusing from the patient's blood into the tissues. Any question about that? And not surprisingly, the Kerby-Bauer test is used very effectively to determine what agent will work well on treating a given bacterial infection. It's not usually used when a patient comes into a hospital, but if the first treatment doesn't work, they frequently will, do the Kerby-Bauer test and then look for another treatment to work. So let's talk a little bit about antibiotics. They're bactericidal if they kill the bacteria, and the bacteriostatic if they inhibit the growth of bacteria. In general, bactericidal antibiotics are preferred because you kill the bacteria cell, you just don't prevent them from growing. Okay, any question about that? Unfortunately, not all antibiotics are equally effective against all bacteria, and that is why we sometimes use this test where we put antibiotics in Dist and then put them on the bacteria to see how effective the different antibacterial agents are on this given bacteria. And that is known as the Kerby, Bauer, Dist diffusion test, sometimes just called the Kerby, Bauer test. So let me blow this up a little bit. So you would think that the bacteria is more susceptible to this antibiotic than it is to that antibiotic, right? But in reality, you have to measure this zone of inhibition and then look it up, because this could be a very small antibiotic which diffuses greater through the agar than this antibiotic. But generally speaking, if you have a large zone of inhibition, the antibiotic is likely, the bacteria will be likely sensitive to that antibiotic. Okay? But if you look at these and these and these, there's either no or little zone of inhibition. So the bacteria are definitely resistant. Well, actually that has a small partial inhibition right there. So let's go to that one and that one. That they have a small zone of inhibition or no zone of inhibition. And if you look at that one right there, there's a haze in there, but there is a definite zone of inhibition, and it's just that there's growth growing within that zone of inhibition. It's not totally clear like that one, and I'm not sure what's going on there. I'd have to look at it. It might be the haze is just on the bottom of the petri dish, or it could be there's some bacteria growing in there. It's hard to tell from that picture. So to run the Kerby-Bauer test, you need to standardize your test. Let me shrink that down so I can see all of that, or at least most of it. You standardize it so that when you do the Kerby-Bauer test and two different people in the world in different places run it, that you will get similar results. Like you don't want to run the Kerby-Bauer test and, I don't know, penicillin G gives a very large zone of inhibition the first time, and someplace else in the world, like at Rome, Italy, the Kerby-Bauer test gives a very small zone of inhibition. So you want to keep the variable standard so that your results will be similar. One way to do that is you always inoculate the nutrient broth from several bacterial colonies isolated on non-selective media that is no more than 24 hours old to grow up your nutrient broth. And then you only grow the broth a little bit so that the bacteria growing in the broth is in logarithmic growth. And this won't mean too much for you, but you grow the broth until the turbidity in the media is 0.5 McFarland standard units, and that's approximately 10 to the 5th cells per mil. That's not very heavy. If you were to grow this bacteria till it was confluent, meaning you can't grow it any further, you'd probably have something like 10 to the 7th organisms per mil. Okay, so they're only grown for a little bit, and that's less than a day that you're growing it in the liquid culture. You inoculate at room temperature a Mueller-Hinton agar plate, so we always put the bacteria on a Mueller-Hinton agar plate. It's always this media, because different media can affect the zone of inhibition. And the Mueller-Hinton plate is always supposed to be between pH 7.2 to 7.4. And this is a mistake here. I don't know why that's 4 millimeters. That should be, the auger should be a depth of 5 millimeters. And I can't correct this. See, I'm clicking on it and I can't correct it. You always have the auger depth the same because the thicker the auger, the less the antibiotic will diffuse through the auger. And the thinner the auger, the more the antibiotic will diffuse. So we always use a plate, a Mueller-Hinton auger plate, that is 5 millimeters deep. And like I said, I would correct that, except I can't. Because I click on it and that must be a picture. And whoever got this and made this picture, they made a mistake. That should be 5 millimeters. We don't need to worry about the diameter of the plate, but you always use a standard plate, and it depends how many discs you're putting on the plate for what the size of that is. But that's the size of the Petri dish. And then you use a sterile, non-toxic swab to spread the plate with the bacteria that you've been growing in liquid culture over the plate. All right? Any question about that part? And then you let that plate dry for about 5 minutes before you put the antibiotic disc on. And you make sure the discs are plated at least 24 millimeters apart, away from each other, and no more than 12 discs on a 150 millimeter plate. And here it is right here. You add the antibiotic disc 5 minutes after you add the bacteria, but not longer than 15 minutes after you add the bacteria to the plate. And then you incubate the plate in a regular incubator, I mean you don't give CO2, and you let the plates grow for 16 to 18 hours at 35 degrees Celsius. And you add the plates no longer than 15 minutes after adding the antibiotic discs. Okay, so that's to highly standardize the test to make sure that wherever you are in the world, who's ever doing the test, that their results will be similar to everyone else who does the Kerby Bauer test. Any question about any of that?
How is the turbidity measured?
It's the same as we do a cell count. We talked about that in the previous lab. You use a spectrophotometer, and then you're measuring the amount of light coming through the media. And I'm not entirely certain how many 0.5 McFarlane units are, but that is the turbidity measure for about 10 to the 5th bacteria cells per mil. Okay? All right, so after you grow up the plates, you then measure the size of the zone of inhibition, and you can use calipers or a ruler to do that. And then you look at the zone of inhibition, the diameter, and compare it to a chart, and you say whether it's sensitive or resistant for what it is in the chart. And each bacteria with each antibiotic will have a different measurement for what's sensitive and what's resistant. And there's an intermediate, and that's the size between sensitive and resistant. And let me see if I can show you something. So we'll be talking about that. I got to come back now and talk about that. But I want to show you the chart here. So here we have the measurement of the zone of inhibition, which it's showing you. Boy, you can't really see in the picture, but that's the zone of inhibition. And then they're measuring it here and here. How you measure this is you don't measure it as 10 or 9 millimeters by this right here, because if you notice, this right there isn't measured. You're measuring it from where this 0 is. So this is measured as close to 15 millimeters. It's not measured as 10 millimeters. And I've had students before who didn't seem to understand that. So I'm telling you, you measure that for where that 0 is. And for some reason on this caliper, they don't have the measurement starting here. So they add this to there, and I don't know. That's just how you measure it, okay? Any questions about that? So let me shrink the size of this so you can see it better. So with the antibiotic ampicillin for E. coli, if the zone of inhibition is 13 millimeters or less, we say that E. coli is resistant to the antibiotic ampicillin. Any questions about that? And if the zone of inhibition is 17 millimeters or greater, then we say E. coli is sensitive to the antibiotic ampicillin. So you go look up on a chart for E. coli and the different antibiotic, and it tells you the zones of the inhibition, and if it's this size or smaller, it's resistant. If it's this size or larger, it's sensitive. And then in between this size and that size, meaning 14, 15, and 16 millimeters, they say it's intermediate. And most species have an intermediate. With Staphylococcus aureus, there is no intermediate. If it's 29 millimeters or greater, then it's sensitive, Staphylococcus aureus, to ampicillin. And if it's 28 millimeters or less, we say Staphylococcus aureus is resistant. But most species actually have an intermediate. All right, any questions about that? So that's how you look up in a chart and then measure the zone of inhibition and see whether it's resistant or sensitive. Okay? Any questions about that? All right, I was talking there. I think I'm done talking about the standardizing. So when you're measuring the zone of inhibition, it's generally best to measure the zone of inhibition from more than one angle, meaning sometimes it's not a perfect circle, and so you take the measurement here, and you take the measurement there, and you might even take a third measurement, and then you take your average diameter, and that is your zone of inhibition. You notice that you are counting the paper disc when you're measuring the zone of inhibition. Okay? If there is no zone of inhibition, you do not say that the zone of inhibition is 6 to 7 millimeters, which is the size of the paper disc. It's actually recorded as 0. Okay? So there is no usually 1, 2, 3, 4, 5. It's always going to be a zone of inhibition of 6 or 7, and record that as 0. Okay? Any question about that? Did I confuse anyone? You do measure the disc when you take your zone of inhibition. All right, know the terms here. Antibiotic, a chemotherapeutic agent used to treat bacterial infections. We already talked about antiseptic, bactericidal, bacteriostatic, disinfection. Excuse me, we talked about disinfectant. A disinfection is when you're referring to the removal of some or all pathogenic organisms from a surface. So like when you disinfect your toilet, you're undergoing disinfection, and it may not remove all organisms, meaning disinfectants, no matter how strong they are, they often do not kill endospores. If you do kill everything, then we call it sterilization, the process that effectively kills or eliminates all transmissible agents, including endospores. Endospores generally are the hardest thing to kill. And in the media, in the lab, how we get sterilization is we usually put things in an autoclave. There are different ways you can sterilize things. Like you could put something under UV light for a long period of time. That will sterilize the surface, but obviously underneath that you'd have trouble. You'd have to turn it over to sterilize the other size. All right, any questions about this lab material? So go ahead and do the lab. You're not going to physically perform any of these activities, but you should know how to interpret the effectiveness of antibiotics using the Kerby-Bauer Dist diffusion test. You're going to watch a video on preparing a spread plate for use with the Kerby-Bauer test, and then review the steps for using aseptic technique for making a lawn of bacteria growth. I don't think I'll talk about that. You can read about that. And then use words to explain the spreading pattern that we'd use to obtain confluent bacterial growth on an agar plate. And then you're going to be looking at the evaluation of antibiotic effectiveness. You will be looking at some clips, 2 movies or videos, preparing a culture for Kerby-Bauer Dist diffusion, and then interpretation of the results of a Kerby-Bauer Dist diffusion test. And then you'll be doing the virtual lab of the Kerby-Bauer test. Any question about any of that? Remember, you're going to be using standards, that various standards are used to do the Kerby-Bauer test. And it looks like we're going to be using, at least you're going to use virtually E. coli to do your Kerby-Bauer test. And then go through and give the results. Like here is ampicillin in the zone of inhibition. Tell me the measurement of that. And the same with these other antibiotic discs. And then take a look at the chart here and read them up. Like for ampicillin, it's looking like it's close to 15. You could say it's slightly less than 15 millimeters. And if we look on the chart, where is 15 millimeters for ampicillin for, is that E. coli we're looking at there? Well, I assume that's E. coli here. It doesn't say it directly, but it might on the questions. So if it's 15 millimeters of E. coli for ampicillin, is E. coli resistant to ampicillin or sensitive to ampicillin? Come on, somebody can answer. Here we go. 15 millimeters ampicillin for E. coli. Is it sensitive to E. coli or ampicillin? Is E. coli sensitive to ampicillin or resistant?
Sensitive.
You want to take another guess?
Intermediate?
Yeah, it's intermediate. 15 millimeters would be intermediate. Thank you, too, for guessing. All right, so you take the measurements here and then compare them to the chart, and then you fill in this table I think in the worksheet you'll have to give your answers underneath the table because you can't write in this table. Well, I'll take a look at that worksheet later. And then answer the questions in the worksheet. Okay? Any questions? If not, go ahead and start working on the lab. I will be here till, is it 3:45? Is that right?
Yes.
Yeah.
Okay, I'll be here until 3:45 to answer any questions. Yeah, you can't write in the table your answers. So when you're filling in the table, you'll have to write below the table. And just add your answers below the table. Come on.